Yeah, in the interest of making sure we cover everything, I'm going to at least begin specific immunity right now. And all right, so far everything we've been doing has been this innate immunity. Again, where it's not looking for a particular thing. It's looking for either things that just don't look like a human cell in general, or sometimes it's just a barrier. You know, the other side is the specific, also known as adaptive immunity. This is the one that we are going to be looking at now. And then we talked about mediated by lymphocytes. But let's start out by just kind of giving its basic characteristics. Like what are the hallmarks of specific immunity? And the main thing I've been talking about up to this point is that it is antigen specific. It's recognizing a very particular shape. So right, and it's kind of showing that example like where a macrophage eats something and then presents a particular identifying marker from that thing that it ate and tells specific immunity, this is what you're looking for. Again, with the coronavirus vaccine, it's priming your immune system to look specifically for something that looks like a coronavirus and nothing else. So it's only going to be about a specific thing. Maybe I should. Antigen stands for antibody generating. You know, and it's the thing that's kind of generating the antibodies. Like, let's say here's a little critter. That's the antigen, and it's got, you know, specific shapes that you can use that distinctively are from this thing. You can actually have more than one kind of recognizing shape for a particular antigen. But let's not go there as much. But yeah, you can have more than one type of antibody, for instance, that recognizes this little red devil thing, right? Because you could recognize the horn, or you could recognize the claw, or you could recognize the tail. That would still give you an identifying marker for that particular antigen. Again, like when you read about monoclonal antibodies, those are antibodies that are cloned from each other that are looking at a specific antigenic determinant site, like one little piece that is always the same. But for the most part, let's just think of antigen as this thing that has a specific shape that you can create some complementary shape that's going to bind and recognize it particularly, right? If I have some antibody that recognizes that little horn, that's going to take this guy out. But if my like coronavirus comes in, you know, with its spike proteins, you know, these are not going to bind to the spike proteins there, but they will bind if this dude comes in, right? So that's when we say antigen specific. It's only going to be targeting one particular target with a particularly shaped kind of marker on it. So it's antigen specific. 2, it is systemic, meaning that it doesn't matter where your body first encountered the antigen. Once this gets rolling, it is going to be protecting you anywhere in your body that your lymphocytes encounter that particular pathogen, or that antigen, I should say, right? So maybe you like got cut on your finger and that's where the bacteria first came in. But once this specific immunity gets ramped up, it doesn't matter where in your body you encounter that particular antigen, you're going to have cells that are primed to recognize it and take it out. As opposed to when we're talking about like some localized infection that's getting walled off and everybody just in that particular battle. That's not setting things up all over your body. That's just wherever it's encountering the thing in the moment. And 3, super important, has memory. Looks like, let me write that better. So we're going to see, once you set up a specific immune response, one of the things that gets created are these things called memory cells. So you're going to have these cells now that are waiting in the wings for the rest of your life, hopefully, that remember that shape of that antigen. And if you ever see that exact same thing again, your body is going to say, oh, I've seen that, and I don't have to do all of the ramping up that I normally would do, and I can just get a really fast, quick immune response and knock it out before it ever has a chance to cause problems. So, for instance, like if I'm going to do some kind of like a generic thing of like y-axis is going to be immune response. You know, whatever that means, like getting everything ready so you can fight whatever. And here is like, you know, antigen encountered. Let's let this be in days. So this is like, you know, 0, 1, 2, 3, 4, 5, 6, 7, 8. So we're looking over the course of days. When you first encounter some antigen, it first is going to have to get eaten by a macrophage and presented to the lymphocytes. And the lymphocytes we're going to talk about go through this clonal selection. And then those clones are going to have to multiply and do what they do. This takes a while to ramp up. Oh, too high. It takes a while to ramp up. And then eventually comes back down. So in this period where the immune system is ramping up, the pathogen has a chance to multiply and get a foothold and make you sick. You know, eventually your immune response has really kicking up here and you fight it and you knock it back and you get better. But in the meanwhile, you got sick. But in this process, so this is like first encounter. The first time you encounter this thing, it takes a while to get this whole response going. In the meanwhile, the pathogen had a chance to make you sick, but then you got better. But you made memory cells in that process. So now, kind of squirreled away in your body are these cells that recognize this thing if it ever sees it again. So now, let's have it happen again. The antigen is encountered again, except this time it's going to be our second time. This is going to be our second encounter. And what you have is a much faster, stronger response because you've already got like all the templates in place. You don't have to like build everything from scratch. What this means is you're probably going to knock it out before it ever has a chance to get a foothold. Now, this is what we call immunity. Immunity is when you have memory of that antigen. And so if you ever see it again, your body has a much faster, stronger response to it, and you are able to knock it out before it ever has a chance to make you sick. So that's all immunity is. So with the the COVID-19 vaccine, it's basically that shot makes your cells make part of like an antigen. It doesn't make the actual virus, right? What it does is, the coronavirus is this thing with these really distinctive spike proteins on it. And this would be like the main thing that actually makes you sick. You don't want to put that in your body. But that vaccine basically kind of tricks your muscle cells into just making these spike proteins, which your immune system recognizes as something weird, but they're not going to make you sick on their own. They have to be part of the virus as a whole to make you sick. So you have these muscle cells making spike proteins, and that's kind of basically going to be the first encounter with this antigen, with the spike protein. I want to be red. So you have this first encounter. You might feel a little off because just the process of having an immune response makes you feel kind of malaise. But you're not getting COVID because there was no actual coronavirus in the whole thing. But you made memory cells that recognize this spike protein. So if an actual coronavirus ends up in your body, then we're at this pace. We're at the second encounter with that spike protein antigen. And your immune system says, oh, I know this thing. You're boom. And take it out before you ever get COVID. Or if you do get COVID, it's going to be very mild. You're going to maybe have a little flu instead of ending up on a respirator and dying. So does that make sense? And so that's a vaccine. Is just giving somebody the antigen, but hopefully not in a pathogenic form. So your body will have memory now for that thing. If you ever see it again, you'll have immunity. There's different kinds of vaccines. You have if the actual thing looks like this, you can have a vaccine that's just a part of it, kind of like what we're doing with the COVID vaccine. You can have a vaccine that is a killed virus. That's the virus, but is not actually pathogenic. Or you can have attenuated versions of the virus, like it's kind of alive, but it doesn't really multiply as virulently as it would normally. So there's lots of different versions of vaccines. You can have parts and pieces of the antigen. You can have killed viruses, like you can have kind of these viruses that are alive but are kind of low. So lots of different versions. Obviously, particularly with kind of a killed virus, there is always like the danger that there's going to be some mutation that's going to make it pathogenic again or something. So the risk of that kind of thing is not zero, but it's really, really low. And compared to the larger societal benefits that you're getting about, like when I was taking my physiology classes, I had a few instructors who just remember, like at the end of summer vacation when they were kids, you're always going to see which of your friends ended up have polio. Who was going to be really sick or maybe paralyzed or limping the rest of their lives. That was just a given. Like the end of the summer season, somebody has got polio. And now we have, vaccines. Nobody gets polio anymore. Um, Vaccines obviously are only as good as the being able to recognize the shape. So some viruses, like the cold virus, mutates its outer protein shell pretty rapidly. So even though you have memory for the cold virus from 2019, the cold virus that comes around in 2021 actually might have mutated enough that it looks different enough that it slips under the radar and damn it, I got a cold again. Although I have to say, nobody I know, since we've all been doing this like shelter in place and washing our hands, has actually, I've never been this long without being sick. It's kind of crazy. I I never quite realized how much just random like exposure to things is what makes you sick. I just figured it's going to happen, but obviously I'd rather run around and have fun and get sick once in a while than do this, but still. Um, Other things about vaccines. Why is it called vaccine? It's a weird name.
Doesn't it come from cow?
Yeah, it's just vaca. It's like Spanish for cow, because some of the original thoughts on how vaccines work was during smallpox, and they realized like that the milkmaids were not getting smallpox, but they had this milder thing called cowpox, which was not fatal, but was similar enough to smallpox that it actually primed the immune system. So if it gave them immunity to smallpox because the antigen was similar enough. So they ended up using that cowpox, virus as a vaccine, basically making people sick with this milder version that would then prime their immune system with memory cells that would keep them from ever getting the more virulent smallpox. But yeah, so vaca is just um can also talk about active versus passive immunity. So active immunity is when you have personally encountered the antigen and have had an immune response and make memory cells. Right, and this is going to give you long-term protection because you've got your memory cells. So if you ever see this again, you're going to take it out. Sometimes though, you want to give somebody immunity to something, you want it kind of instant. Um, so you can just inject somebody with pre-made antibodies towards whatever the antigen is. For instance, when I was in grad school, I ended up getting Hep A from a trip down to Baja. Went diving and ended up getting so sick. And all of my roommates all had to get shots of, they call it antibodies, or also I mentioned gamma globulin, immunoglobulin, antibodies. Those are all synonyms. Right? So all my roommates got shots of gamma globulin that had the shape that would target the hepatitis A virus. So they were instantly immune to getting Hep A. So we could all hang out together, and they weren't going to get sick from me. Um, But what's the difference between, so it's nice because it's instant and you can just get it with a shot. What's the downside of it?
You're not forming memory?
Exactly, you're not forming memory. So there's, no memory. Eventually, those antibodies cleared out of their system and then they were vulnerable to hepatitis A again. Whereas, I can never get it again because I have memory cells and my body will recognize it and will take it out. Right? I mean, like, obviously, the other thing is you can get active immunity either by getting the actual disease or by a vaccine. The vaccine gives you active immunity as well because it's triggering your immune system to go through its whole process and make memory cells. Um, so that is active versus passive immunity. And again, the basic idea of immunity. Immunity is second encounter with the same thing, due to your memory cells, you get this really fast, strong response. So you really aren't going to get sick like you would have normally if you hadn't had immunity. All right, so what I will do, I'm going to introduce the two divisions of the, I'm not going to go all the way finish this because I want to make sure I don't burn you all out on it, but I want to kind of introduce the two main divisions of specific or adaptive immunity. But again, remembering whether or not we're looking at antibody mediated or cell mediated specific immunity, we are always talking about something that is antigen specific, systemic, and has memory. But now what we're going to do is further drill down on the two main divisions of specific or adaptive immunity. There we go. So one is going to be called humoral or antibody mediated. The other one is going to be called cell mediated. From their names, you get a sense of kind of the difference. The way that the humoral or antibody mediated specific immunity destroys their targets is through the use of antibodies. And again, we met antibodies when we were doing the blood typing, those little sticky wickets that recognize a particular shape and grab on. One of the ways they help destroy their target is through agglutination, which you saw in the blood typing lab. They make them all stick together and they can't get all in a big glop and the white blood cells can come in and eat them. It's also going to be fixing complement I was talking about and some other stuff we'll talk about. But again, the things that are actually kind of attaching to the target and destroying it are the little antibodies. And again, antibodies aren't cells, they're just little plasma proteins. We talked about they have that particular variable region. So that's antibody mediated. Cell mediated is more like those natural killer cells. They are going to be killer cells, killer T cells, that go in and recognize usually cells that have been infected with stuff and take the whole cell out by poking holes in them. So, again, these poke holes in the target. So, just like we saw with the natural killers, you're going to insert those perforins into the target, as well as then like send in these other enzymes that help destroy the thing. So that's like one of the big differences. The other big difference between them is that they use a different subtype of lymphocytes. Cell mediated has lymphocytes that are maturing in the thymus gland, and they call them T lymphocytes. Humeral uses B lymphocytes. These mature in the bone marrow. Often they call them just T cells and B cells. But if you want to be more formal, it's like B lymphocytes and T lymphocytes. And part of this maturing, we'll talk about more, but it's making sure that you don't have any of these that might recognize your own tissue as its target antigen, right? Because it's a disaster if all of the stuff that this does to kill invaders gets targeted on your own tissues. Again, that's what we have going wrong in all these autoimmune diseases like rheumatoid arthritis or multiple sclerosis, where you're attacking your own myelin. So it's really important that you're super careful about taking out and removing any potential connection, any cell that might be primed to recognize your own tissues. What else? Both of them are going to make memory cells. There's going to be memory T cells and memory B cells at the end of everything, because, like I said, that is a characteristic of all adaptive immunity. So we'll see different populations. We'll see B memory cells, T memory cells, but then we'll see the B cells, the plasma cells that make the antibodies. In addition to the killer T cells, there's going to be helper T cells and blah, blah, blah. Let's see what else I want to say here. So we're going to return. We'll speak more on Tuesday. I'll talk a little more about the differences between these and some of the subpopulations of the different lymphocytes. But I kind of want to just lay out this big picture. For specific immunity, there are two branches. One branch that uses antibodies, one branch that uses these killer T cells. Killer T cells poke holes in their target. Usually it's like virus infected cells that are getting taken out. Humeral, on the other hand, is using the antibodies, those little sticky wickets that attach to things and then either gum them up or make the complement cascade happen or something like that. Okay, I'm going to cover one more thing and then we'll take another break. Major histocompatibility complex. So this is a membrane protein on every one of your body cells, I think except for like RBCs. And remember histology and anatomy? What is histology? Study of?
Cells?
That would be cytology.
Oh,
Tissues?
Tissue, study of tissue. So it's basically tissue compatibility. So this is an identifier saying that this cell is your tissue. This cell belongs to be part of you. So one part of this membrane protein is it's kind of like the barcode that says that this cell is part of you. You know, this is the reason why organ transplants are really tricky, right? You know, let's say, you know, I see Raina there, so I'll say Raina. Raina is there and desperately needs a kidney, and I'm thinking like I can survive with one kidney, so I'm gonna give her my kidney. There's a good chance that it doesn't matter how much I want to, that her MHC is different from my MHC, and if my kidney ends up in her body, her immune system is going to say that MHC is wrong. This is something that doesn't belong here. Let's attack it and kill it. You know, the only way that organ transplants work is if you find somebody that's really closely genetically related. Hopefully, you know, more likely it's going to work if it's someone in your family, or you know, they'll do all sorts of genetic typing, and you know, they can find something where one person's MHC is close enough to another person's MHC that you're not going to have as strong of a reaction. But even then, you know, the recipient is going to be on immunosuppressant drugs for the rest of their lives and be at way higher risk of, like, you know, dying of a common cold or something. So MHC is kind of the barcode, the little tag that says this cell belongs here and it comes from my, you know, my original genetic material. So that is, so just cell ID. The other thing that MHC, and in particular, there's two kinds. The one that's most common is called MHC. Most membrane protein almost all is MHC1. MHC1 has one other property that make it really cool. So as obviously it's a membrane protein, so it's just a protein being assembled in the cell and then getting inserted into the membrane. As it's getting assembled, a random peptide fragment, like how many? It's like, I have something, it's like 7. How many is it? Or do I even have it? It's somewhere around like 7 or 8 amino acids long. Hold on, let me nab it. Oh, there it is. Yeah, 8 to 9 amino acids long. So I'm gonna put some little protein, some little peptide. These are amino acids, right? An endogenous peptide fragment. So it gets displayed here on the MHC, MHC1. And again, this was just, there's little random things floating in the cell. And as this thing is getting assembled, it grabs some little fragment, some schmutz floating around in there, and puts it on there as part of the protein that is now on the outside. So why is this useful? All right, so kind of if we go back to, remember I was talking about like these killer cells. Let's say this is like a killer T cell. And he's got his particular antigen that he's looking for. He wants to see some virus. He wants to see if, he's got his little like APB out for this thing. It's like, I need to look out for this thing because this is a thing that is the antigen I'm trying to get. And he's looking, but if you're a cell infected with this virus, the virus is inside, right? The virus that's killing the cell is inside the cell. It's not on the outside. So this killer T cell is looking around. He wouldn't be able to even know that this virus is right in front of his nose because it's inside the cell. It's not on the outside of the cell. But MHC1, as it's getting made, it's taking some random piece of whatever is in the cell and displaying it on the outside of the cell. Right? So if this is just some random stuff, it doesn't matter. But if this cell is infected with the virus, the chances are there's going to be like, oh my God, there's a little piece, there's a little horn from the thing. So now my killer T cell is looking around and there on the MHC1. He's like, bingo, holy crap, this thing is in trouble. And then, sad for the cell, but happy for your body. He's going to take this thing out and neutralize it before it just ends up being a big bag of viruses that is released into the body. So that MHC1 is a way. It would be like, most people have seen Alien, right? If I had a little thing saying, here's a bit of inside me, and you looked and it was like a bit of liver. It's like, not to worry, should be a liver in there. Oh, here's a little piece of intestine. Nothing to worry about. That should be a piece of intestine in there. Oh, here's some weird little alien with weird teeth. And it's like, okay, there's something wrong. There shouldn't be some weird, crazy alien inside me. I am obviously about to have my chest burst open and then like cause even more problems for everybody else on the spaceship around me. So it's better just to kill me now. Right? So that's kind of what that MHC1 is doing. MHC2 is only found on the antigen presenting cells. I'll just do that really briefly because I'll write it out just for completeness. Right. Remember, I talked about how these antigen presenting cells eat things and present that little piece to the rest of the immune system. MHC2 is the mechanics of that, right? It does not present a little random piece of the inside of the cell. It specifically it's basically if I am, heck, I want it to stay red. So my antigen presenting cell comes and eats this thing. And now it's inside. And now I'm going to make this special MHC2 that just has a special little piece of this thing that I ate. So rather than displaying some random thing from inside the cell, MHC2 displays something, a piece of something that the cell phagocytosed and ate. That's why exogenous, something that came from outside, not some random thing that came from inside the cell. Does that make sense?
Yeah, and the endogenous peptide fragment is you said the 8 to 9 amino acids that came from inside the cell.
Correct. So in MHC1, there's just some random fragment that started inside the cell and then got assembled into MHC1. So most of the time, if the cell's healthy, it should just be some normal thing. It shouldn't be anything weird. But if the cell has been attacked by viruses, then it might actually be a piece of the virus that is now kind of infesting the cell.
So then why would an MHC2, why would there be an exogenous peptide fragment being presented to the cell?
Because these are specific, it's because these are, they're special.
Activate the immune response. Right?
Yes, their job is to say, this is the thing I just caught and ate, and I'm going to use this to now, basically go to headquarters and say, we need to create an immune response. Like if we go back to this place right here, antigen encountered,
Okay.
that is that moment. It's like this thing eats this thing, is displaying it, and is now activating the other lymphocytes to create the whole response here.
So it's kind of like presenting it to the cell for the cell to decide, how it's going to react.
Yeah, so MHC2 is used to present the antigen to the other lymphocytes to tell them this is what has just been captured and this is the template that you should use to make all your antibodies or whatever.
11 a.m.
All right, so that's that. Boom, boom. Yeah, I mean, people use MHC as just a measure of genetic how closely or farther away you're related genetically. Like I think I told you there was that like pheromone study where they were like looking at things that are taken in by your nose, but you don't detect a smell, but actually influence kind of things. And they like had people wear t-shirts and then put the t-shirts in a box, and then they had other people smell the t-shirts in the box, and some of them were like kind of nice and that's pleasant, or other ones like you had to pick which one was more or less pleasant. And they found that there was a major correlation with MHC, with whether or not, like, if your MHC was too close to the MHC of the person who was wearing the t-shirt, it was like, yeah, nah. And when there was more distance, then it was more like, oh, this is comfortable, this is nice. So they're thinking that, there's just something happening. People are actually exchanging molecules when they're just sitting across from each other, that's already giving each other information about how genetically related or not they are. And if you're too genetically close, you're probably not going to be as likely to take the next step and mate with them or something ultimately. Whereas if there's a little more genetic distance, then there's more likely to be a good genetic diversity between the two of you. So it's not like, oh, I must have this person, or like, ooh, but there is something subtle that's going on, just based on little molecules we exchange across the air without even touching each other, about how closely or farther away we're related genetically.